Biological macromolecules. Let's look at molecules in general, and then we'll talk about what makes an organic compound, and then discuss the four major macromolecules: carbohydrates, lipids, proteins, and nucleic acids. When we study molecules, a lot of them are largely made of carbon, and when a molecule is based in carbon, we call it an organic compound. There are a few different ways to look at molecules, and what you see in this picture is a carbon skeleton model. You'll find a lot of different pictures. The letters, C stands for carbon and H is for hydrogen. Those letters are just telling you what the elements are. Those are the symbols from the periodic table of elements. The lines between the individual elements signify their bonds. One line between two elements means they share one electron, so it's a single bond. Two lines, like in the top right-hand corner, in a molecule means a double bond, so they share two electrons. The molecules with only carbon and hydrogen in them are known as hydrocarbons. We study hydrocarbons a lot because they carry energy. Gas is an example of a hydrocarbon, the gasoline you put in your car. It's a hydrocarbon. A few chemical groups, few chemical molecules, are the key to the functioning of biological molecules. Here you see 15 major chemical groups called functional groups. If you ever take organic chemistry, you'll get to know each of them very well. There are 5 that we're going to touch on over the course of the semester. The first is a hydroxyl group. When you add an OH to something, we call that a hydroxyl group, and that thing becomes an alcohol, technically speaking. A carbonyl group is a group that has a double bond in it, and it has an OH on it as well, and they make aldehydes and ketones. These are very important for metabolism. A carboxyl group is a hydroxyl and a carbonyl group added together, and they act like acids. So there's a lot of H pluses in the solutions that they're in. Amine groups have a nitrogen in them, and they act like bases, so OH is in them. And phosphate groups are a group that kind of stand out because they're very popular because they hold on to energy, and they're constantly involved in reactions, and actually basically every reaction that happens on our planet and requires energy. When we look at molecules in chemistry, we talk about the making and breaking of chemical bonds. Single units are often called monomers, and multiple units linked together are called polymers. When you add monomers and polymers together, you make macromolecules, and they're built with dehydration reactions. The OH that hangs off the end of one molecule, it's really common for that kind of structural balance to be there, and there's usually a hydrogen hanging off of a second monomer. A water molecule will actually form when that OH and H get close enough together. The polymer will link up and the water will leave. If you do this enough times, you end up with the macromolecule, or a really long chain of repeating units. To make something solid, you remove the water, you dehydrate it. So this is a dehydration reaction. It makes sense if you think about something in cooking. Removing the water makes it more solid. If you want to make something less solid in your kitchen, your soup is too thick, you add water to it. Do you want to break up a polymer? Hydrate it. Add water. And it looks like this. You get the OH back on one side and the hydrogen on the other side. The monomers will slowly break off from one another piece by piece, and you have a bunch of single units rather than a long chain. When we study biological macromolecules that are important, we'll look at a lot of these monomer, polymer, macromolecule kind of setup. The 4 biological macromolecules that we're going to look at are carbohydrates, lipids, proteins, and nucleic acids. Carbohydrates are polymers of sugar. A simple monomer is called a monosaccharide. One commonly discussed and seen in biology is glucose. If you just change the location of a bond, it actually becomes fructose, but it has all the same building blocks. 2 monosaccharides together are called disaccharide. A really common disaccharide in biology that's discussed is called maltose. It's a food for a fly that we'll study in genetics. If you add a whole bunch of disaccharides together, you get a polysaccharide. We'll talk about starch, glycogen, cellulose, and chitin over the course of the semester too. You go from monosaccharides to disaccharides to polysaccharide using the dehydration reactions that we studied before in the monomers, polymers, and macromolecules? Look at this reaction and decide a couple of things. Do you think this picture is showing you a dehydration reaction or hydration reaction? And once you figure that out, tell me, what would you call the molecule at the top and what would you call the molecules at the bottom? You should be able to attach some vocabulary terms to these concepts. The second biological macromolecule we'll discuss are lipids. Lipids are fats with fancy names. Lipids are incredibly diverse molecules that are classified together essentially because they don't like water. They differ slightly from the carbohydrates because they're neither macromolecules nor polymers built by units. They do act in kind of large groups, though, as part of a team. Lipids are made from two smaller molecules, glycerols and fatty acids. You can see in the larger image here that we can add on to those units to specialize the lipid. The basic form, that's the little guy on the right. The head is hydrophilic in nature. It loves water, chemically speaking, and the tails are hydrophobic. They hate it and band together to shield themselves from it. Lipids make up cellular membranes and are considered long-term storage molecules. We group fatty acids into two major categories, saturated and unsaturated fats. Unsaturated fats have kinks in their tails because they contain double bonds. They're liquid at room temperature because of those kinks. Saturated fats only have single bonds and therefore they have very straight tails. They can pack really tightly and are solid at room temperature. We think of unsaturated fats as healthy fats because they're more easily digested and are therefore more quickly removed from the body. We'll study these molecules really closely when we talk about cellular membranes. Proteins are the third biological macromolecule. They are made up of small building blocks called amino acids, and their functions are endless. We'll look at proteins in a few different ways, and what you see here is called a ribbon model. Notice the ribbon curl shapes and the green kinks that kind of look like pleats. These pictures, or these pieces, are incredibly important as the structure of a protein is tied directly to the function of a protein. We know this because when we denature a protein or unravel it with heat, it stops performing its job. These are what those individual units look like. There is an amino group on one side, a carbon and hydrogen in the middle, and then a carboxyl group off to the other side. Notice the blue box that surrounds that R. The R group is a variable for all 20 of the different amino acids, and the R group is essentially what defines that amino acid. So this whole piece is an amino acid. All of the pieces of amino acids always look the same, with the exception of the R group. Dehydration reactions make polymers of proteins as seen here. Two amino acids can react with one another and a molecule of water is removed and a new bond is made. The bonds in the proteins are referred to as peptide bonds. It's just another piece of vocabulary that allows scientists to talk more specifically about their specialties. Anytime you see peptide or polypeptide, you're learning about a protein in some form or fashion, and that vocabulary just allows you to be more specific. Proteins have four major levels of structure. The most basic structure of a protein is the primary structure. It deals only with amino acids as you would read them in a line. The secondary structure deals with alpha helices and beta pleated sheets, so those curls and kinks that I mentioned previously. The tertiary structure is created by R groups interacting with one another, and it's kind of folding all of your curls and kinks into very specifically formed balls. And some proteins, they end their structural advancements with tertiary structure. That's as fancy as they get. The fourth layer of structure in proteins only happens in a few different proteins, or a few different protein families, and that's when multiple individual units need to work together to create kind of some master functional unit. So think about the transformers. Every individual transformer has a different name, but they come together to form Megatron, and if you're not a nerd, I apologize for that reference, but it makes sense even if you only vaguely know your transformers. Here are those levels again. Primary structure of protein is just the amino acids. You read them off name for name for name. It would kind of be like reading primary structure and saying P-R-I-M-A-R-Y. You just read the letters. The secondary structure refers to the alpha helices and the beta pleated sheets. So you have the alpha helices, the curlicues, and the beta sheets, beta pleated sheets, or kind of the crinkle of paper fans that you might have made as a kid. The tertiary structure comes from the R groups interacting with one another and forming a more condensed structure. And the quaternary structure is when multiple polypeptides are associated with one another to create one huge mega functioning protein. The final biological macromolecule are the nucleic acids. DNA and RNA are two types of nucleic acids. The amino acid sequence of polypeptide is actually programmed by a discrete unit of inheritance known as a gene. Genes consist of DNA, nucleic acids, and DNA is translated into RNA, a different nucleic acid, which supplies the messages that are made into proteins. Nucleic acids are polymers of something called a nucleotide. That's what you see here. At the center of a nucleotide is a 5-carbon sugar. Off to one side is a phosphate group that holds energy, and then finally there's a base off to the other side that makes the nucleic acid unique. The differences in RNA and DNA are quite obvious and have big impacts. They're two different sugars at their centers, for instance, and when it comes to the bases, RNA uses cytosine, guanine, adenine, and uracil. DNA, however, it uses different bases. It uses cytosine, guanine, adenine, and thymine. You never see thymine in RNA, just like you never see uracil in DNA. RNA, as you see in this picture too, also exists as an open, single strand of molecules. DNA, far more protected. The bases pair with one another down the middle, kind of this ladder type structure with 2 rungs on the outside. We'll be looking at DNA and RNA in great detail in our study of genetics. First, however, let's look into cells and their functionality as individual units. That's where we're going next in class.